Assalamualaikum warahmatullahi wabarakatuh. My name is SITI and NIZAM. We from AS114182. Today we want to explain about the active transport. Before we start, let me introduce my group's members. The first one is EMYLIA NATASHA. Second one is ATIQAH. And lastly, ADEENA FITRISHA. What is active transport? Active transport is the movement of the substance across the plasma membranes against the concentration gradient. The process only occurs in living systems which are active, producing energy through respiration. Active transport needs the supply of ATP energy. The energy is needed for moving the carrier protein so that they can transport substance against the concentration gradient. The movement of substance requires carrier proteins. The carrier protein receive energy from ATP molecule and change shape and get active to transport the substance contained across the plasma membrane against the concentration gradient. This is example of active transport. The first one is sodium-potassium pump. The second one is cotransport. The third is endosynthesis. And the last one is exocytosis.
Assalamualaikum warahmatullahi wabarakatuh. So I want to present about sodium-potassium pump, an internal protein that exchanges 3 sodium ions, move out of cell with 2 potassium ions that move into the cell. The process of ion exchange against the gradient is energy dependent and involves a number of key steps. So the 3 sodium ions bind to intercellular sites on the sodium-potassium pump. A phosphate group is transferred to the pump via hydrolysis of ATP. Then, the pump undergoes a conformational change translocating sodium across the membrane. The conformational change exposes 2 potassium binding sites on the extracellular surface of pump. Then, the potassium group is released, which causes the pump to return to its original conformation. To this, and they translocate the potassium across the membrane, completing the ion exchange. For the cotransport occurs when active transport of a solute indirectly drives transport of other solutes. Plants commonly use the gradient of hydrogen ions generated by proton pump to drive active transport nutrients into the cell.
Okay, saya akan menerangkan tentang bulk transport. First, bulk transport across plasma membrane occurs by exocytosis and endocytosis. Maksudnya, pengangkutan daripada plasma membrane. Yang kedua ialah small molecules and water enter to leave the cell through the lipid layer of via transport protein. Maksudnya, molekul kecil seperti air memasuki atau meninggalkan sel melalui lapisan 2 lipid atau memasuki transport protein. Yang ketiga ialah for large molecule such as polysaccharide and protein cross the membrane imbued via vesicle. Maksudnya, untuk molekul yang besar seperti polysaccharide and protein akan melintasi membrane secara melalui vesicle. And the last point is bulk transport require energy. Maksudnya, bulk transport ini juga melakukan pengangkutan dan memerlukan tenaga. Yang selanjutnya ialah exocytosis. Exocytosis merupakan proses by which a large substance exit the cell without crossing the membrane. Maksudnya, proses ini akan berlaku sama ada bahan itu. The first point, vesicle Typically derived from the Golgi, fused with the plasma membrane, expel the contents into the extracellular environment. Maksudnya, vesikel ni kebiasaannya berasal daripada Golgi, menyatu dengan plasma membran dan mengeluarkan kandungannya ke sekitaran ekstraselular. Poin yang kedua, the process of exotosis at cerviculum phospholipid to the cell membrane, replacing those lost when vesicle are formed via endotosis. Maksudnya, fresh endotosis ini menambahkan phospholipid vesiculum ke sel membran, menggantikan yang hilang ketika vesikel berbentuk melalui endositosis.
Endocytosis is the process which large substance enter the cell without crossing the membranes. The cells also takes in macromolecules by forming the vesicle from the plasma membrane, and invagination of the membranes forms a flash-like diffraction which involves the extracellular material. The invagination is then sealed off to form an intracellular vesicle containing the material. Endocytosis is a reversal of exocytosis, which is involving in different proteins. There are 3 types of phagocytosis. The 2 main types is phagocytosis, which is cellular eating, and pinocytosis, which is a cellular drinking. And the last one is receptor, which is mediated endocytosis. Now I would like to explain a little bit about the 2 main types of endocytosis. First is phagocytosis. It is the process which solid substances are ingested. It is usually to be transported to the lysosome. In phagocytosis, a cell engulfs a particle in a vacuole. The vacuole fuses with a lysosome to digest the particle. The next is pinocytosis, the process which liquid substances are ingested. It is allow fastest entry via the protein channel. In pinocytosis, molecules are taken up when extracellular fluid is gulped into tiny vesicles. So that is all from me. Thank you.